Welcome to another episode from the Veterinary Research Channel. Today, we will explore a fascinating area of research that uncovers how our cells can defend themselves against bacterial infections using a previously unknown mechanism. This mechanism involves the cellular machinery responsible for protein degradation, known as proteasomes. Buckle up as we delve into the world of antimicrobial peptides and how they can be derived from our own proteins to fight off invaders. In the realm of immunology, antimicrobial peptides, or AMPs, are small molecules with broad-spectrum activity against bacteria, viruses, and fungi. Think of them as the body's natural antibiotics. Many of these peptides are encoded and produced as functional units. However, some lie hidden within larger proteins, requiring processing after translation to become active. Thousands of AMPs have been identified across various organisms, but a systematic exploration of their presence and function in human proteins is just beginning. Researchers sought to understand how widespread AMPs are within the human proteome, which is the entire set of proteins expressed by human cells. They compared a list of experimentally confirmed AMPs from different species with the human proteome and identified 308 potential AMPs within 273 human proteins. These AMPs shared identical sequences with previously reported peptides known to have antimicrobial activity. Interestingly, when scientists looked at the conservation of these AMP sequences across different species, they found that a significant portion, about 28.5%, were more conserved than other regions of the host protein. This suggests that there might be a selective pressure to maintain these sequences due to their functional role in antimicrobial defense. The proteins containing these AMPs included histones, which are involved in DNA packaging, transcription factors that regulate gene expression, and many proteins with functions not directly related to the immune system. These proteins are expressed in various tissues. Some are specific to only one or two tissue types, such as SERPIN1 in the liver, which was not previously known to have antimicrobial activity in humans. Others, like Hox genes, are widely expressed. The best-studied AMPs are highly expressed in immune-related and secretory tissues, such as bone marrow, tonsils, testes, salivary glands, and the small intestine. The location of these potential human AMPs within proteins is also noteworthy. They are predominantly found in structured regions with slightly reduced accessibility. This suggests that these AMPs may not be readily released unless the protein undergoes unfolding or degradation. This observation led to the hypothesis that protein degradation, specifically through cellular proteasomes, might be an unexplored mechanism for AMP maturation. To investigate whether protein degradation could yield protective peptides, scientists artificially cleave the human proteome using predictive proteasomal cleavage sites. This in silico digestion generated a database of approximately 34 million peptides. Since AMPs typically act outside the cell, researchers look for these predicted proteasomal cleaved peptides in public peptidomics datasets of biological fluids. They found that 15% to 27% of the peptides detected in various biological fluids matched those predicted by the in silico cleavage, suggesting that proteasomal cleaved peptides can be secreted into the extracellular environment. To assess the antimicrobial potential of these peptides, a scoring metric was used based on peptide charge, the presence of cationic and hydrophobic residues, and peptide length. Peptides with a score above 5, which is the average score of validated AMPs, were considered potential proteasome-derived defense peptides, or PDDPs. Hundreds of thousands of these peptides, about 1.2% of the total peptide pool, displayed biochemical characteristics of AMPs. Given that proteasomes generate peptides at a rate of millions per minute, cells possess an enormous potential to produce protective peptides under normal conditions. To directly test the hypothesis that AMPs are generated after the breakdown of cellular proteins, cells were infected with Salmonella typhimurium, an intracellular bacterium. Some cells were pretreated with a proteasome inhibitor. After proteasome inhibition, there was an increase in intracellular bacterial infection, suggesting that proteasomes play a role in controlling bacterial infection. Next, the researchers examined the antimicrobial activity of the peptides secreted by cells into the extracellular environment. They observed that inhibiting proteasomes enhanced bacterial growth in the conditioned medium. To confirm that this effect was due to peptides rather than other secreted molecules, the conditioned medium was treated with proteinase K, an enzyme that cleaves peptides. The proteinase K treatment abolished the observed bacterial growth inhibition, confirming that the peptides in the conditioned medium were responsible for the antibacterial activity. To further explore the function of proteasome-cleaved peptides, researchers used mass spectrometry analysis of proteasome-cleaved peptides, or MAPP. This method captures a snapshot of active degradation by identifying degradation products in or near the proteasome. Analyzing degradation product from different cell types under various conditions revealed several previously reported AMPs, such as histidine 3. Most known AMPs are cationic, hydrophobic peptides with lengths between 10 and 50 amino acids, similar to the length distribution of MAPP peptides. These MAPP-identified peptides were also detected in extracellular peptidomic datasets of biological fluids. To validate the secretion of proteasome-cleaved peptides, peptidomics analysis was performed on the secretome, the collection of secreted peptides from A549 cells. Comparing MAPP and secretome peptides, 110 peptides were identical and 383 were contained within MAPP peptides, indicating further processing of these peptides. To identify potential PDDPs, MAPP-identified peptides were scored using the same scoring system mentioned earlier. From over 50,000 peptides, 1,074 peptides with membrane-disrupting properties were selected. 10 high-scoring peptides were synthesized, along with low-scoring peptides as negative controls. LK20, a known antimicrobial peptide, was used as a positive control. The high-scoring peptides were derived from proteins with diverse functions, including PPP1CB, a catalytic subunit of a serine-threonine-specific phosphatase, PSMG2, a chaperone involved in proteasome assembly, DCTN4, a dynactin subunit associated with the centrosome, and DFNA5, also known as gastromin E, which promotes programmed cell death. The antimicrobial activity of MAPP-identified peptides was tested against several bacterial species, including Pseudomonas aeruginosa, Escherichia coli, Salmonella enterica, Staphylococcus hemolyticus, and Micrococcus luteus. These species represent a broad spectrum of bacteria, including both gram-negative and gram-positive types, as well as intracellular and extracellular bacteria. The potential PDDPs induced bacterial inhibition or killing in a dose-dependent and selective manner. The minimum bactericidal concentrations were lower than the minimum inhibitory concentrations, and the low-scoring peptides showed no antimicrobial activity. To explore potential synergistic effects, peptide combinations were tested. A combination of 5 peptides showed additive activity against E. coli, and a combination of all 10 peptides had synergistic activity. However, the antimicrobial activity of peptide combinations against M. luteus, P. aeruginosa, and S. hemolyticus was similar to that of individual peptides. To assess the activity of PDDPs under physiologically relevant conditions, the half-life of the peptides was evaluated. The half-life distribution was near normal across the proteasome-derived peptides, with no significant differences between peptides with or without potential antibacterial activity. There is only a weak correlation between the AMP score and the predicted half-life. The PPP1CB derived peptide had a predicted half-life of 23.3 minutes in blood and exhibited prominent antimicrobial activity against diverse bacterial species. The activity of the PPP1CB derived peptide was further tested against P. aeruginosa, a bacterium known for its resistance to antibiotics. First, a transepithelial-transendothelial electrical resistance assay was used to measure the integrity of tight junctions in a monolayer cell culture. It was found that the integrity of the gut epithelial barrier, represented by a CACO2 monolayer, was disrupted by the presence of bacteria. However, this effect was completely reversed by the presence of the PPP1CB peptide. The potency of the PPP1CB derived peptide was assessed in 2 mouse infection models, acute pneumonia and bacteremia. In the pneumonia model, the bacterial load in the lung was significantly reduced by intravenous administration of synthetic PPP1CB, comparable to the effect of tobramycin. Neutrophilic infiltration and tissue injury were also diminished after PPP1CB administration. In the acute bacteremia infection model, intravenous administration of PPP1CB resulted in a significant reduction in bacterial burden in the spleen. The concentration of PPP1CB used was 5-fold lower than that of tobramycin, though administered via a different route. Furthermore, the PPP1CB PDDPs-derived peptide led to a significant decrease in pathological indicators, such as immune cell infiltration and tissue injury, across multiple organs. Finally, the effect of the PPP1CB-derived peptide on sepsis-associated mortality in mice was examined. The PPP1CB-derived peptide led to a significant reduction in mortality. These results indicate that the PPP1CB-derived peptide may have therapeutic value and highlight the potential of additional PDDPs to serve as a new class of antibiotics. To determine whether targeted proteasomal degradation of the full-length PPP1CB protein could induce antimicrobial activity, researchers first confirmed that knocking down PPP1CB had no effect on bacterial growth. Then, they used the DTAG system, which induces degradation by targeting an exogenously expressed protein to the proteasome. Treatment with the DTGV-1 drug activates the system, leading to the degradation of PPP1CB. This overexpression system generated a pool of PPP1CB degradation products, including 4 peptides that scored above the threshold as potential PDDPs. The upregulation and subsequent degradation of PPP1CB alone were sufficient to reduce bacterial growth by 20% when the conditioned medium was used. To examine the antimicrobial activity of intracellular PDDPs against intracellular bacteria, cells were pre-incubated with DTGV-1 to activate PPP1CB degradation. Activating the DTAG system significantly reduced intracellular bacterial infection. Even though the PPP1CB-derived peptide was detected both intracellularly and extracellularly, these findings support the idea that targeted proteasomal degradation can induce antimicrobial activity. In conclusion, this research uncovers a previously unknown mechanism of cell-autonomous innate immunity, where proteasomes degrade cellular proteins to generate antimicrobial peptides. These proteasome-derived defense peptides exhibit antimicrobial activity against various bacteria and can protect against bacterial infections in vivo. The discovery highlights the potential of harnessing this natural process for new therapeutic strategies against antibiotic-resistant bacteria. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.